Greetings everyone, and welcome to another exciting genetic engineering and biotechnology news webinar. Our presentation today is entitled Quantitating Critical Subcellular Events in Significant Populations Using Imaging Flow Cytometry. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Visualizing discrete biological samples has always presented challenges for researchers, from minimizing distortion to maximizing resolution. Yet having high throughput capabilities to view tens of thousands of cellular events based on their fluorescent and morphological parameters in order to obtain a greater understanding of cell populations was, until recently, something investigators could only wish for. Now, imaging flow cytometry is revolutionizing visual technology by combining the sensitivity and analytical power of flow cytometry with the spatial resolution of fluorescent microscopy. With the aid of imaging flow cytometry, scientists can now begin to answer critical questions concerning the subcellular distribution of various molecules within cells that are critical for disease states. Let's meet our panelists for this GEN webinar, who will discuss their novel approaches for utilizing imaging flow cytometry to tackle the underlying mechanisms of immunology and cancer biology. Our first speaker today will be Olivier Thaunat, Assistant Professor at the Edouard Herriot Hospital within the University of Lyon. Dr. Thaunat will describe his work on identifying the polarized distribution of antigens on B cells in vivo using imaging flow cytometry. Our second presentation will be given by Christopher Parris, Senior Lecturer and Director of the Biosciences Division in the College of Health and Life Sciences at Brunel University, London. Dr. Parris will outline how he utilizes specific features of imaging flow cytometry for the quantitation of DNA-damaged cells, providing a high-throughput method for the detection of gamma H2AX foci. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentation. We'll try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. So let's begin our webinar and open up the floor to our first speaker. Olivier?
Hello everybody. My name is Olivier Thaunat. I'm an MD PhD in Lyon, France. And in my group, we're interested in trying to understand some basic aspect of B-cell biology. What I'd like to convince you today is that imaging flow cytometry is a technology that is really worth keeping in mind whenever you design your own research. And I will try to convince you of that by using some data we have generated in my group. As you probably know, T-cell dependent humoral immune response is the process by which you can generate antibodies against protein antigen. It is a multi-step process that starts with the binding of cognate antigen to the B-cell receptor of B-cell clone. This triggers a signaling cascade inside the B-cell. Before going further, I need to introduce to you a nice tool that we use to dissect this process of B-cell activation that we call synthetic particulate antigen. These synthetic particulate antigen, or SPAG, are basically just polystyrene beads, very small, only 400 nanometer diameter, which is close to the size of a pathogen like viral particles, for example. And not only are they fluorescent, so you can track them, but they also are coated with streptavidin, which allow you to come with any biotin-related antigen and to coat these particles with your favorite antigen. Using this technology, the team of Facundo Batista, where I made my postdoc a few years ago, has shown that the binding of the antigen to the B-cell receptor was triggering a cascade of events leading to the association of the microsignalosome to a molecular motor called dynein. And this molecular motor is responsible for dragging actively the microsignalosome along the microtubules. And this is very important because this is the mechanism by which the antigen is going to be internalized by the B cells. Because all the microtubules are anchored by their minus end to MTOC, which means Microtubule Organization Center, of course, the antigen that has been internalized inside the B cells will not be located anywhere in B cells, but instead will be polarized at the mTOC of B cells inside endosomal compartment, which are LAMP1 positive. Internalized antigen is then processed and presented by B cells into MHC class II molecules at the cell surface. This is really important because this complex made by MHC class II molecules and processed antigen will be recognized by cognate helper T cells that will in return provide the second signal of activation that is crucial for this T cell dependent humoral immune response. It is the combination of these two signals of activation that allow the B cells to differentiate into antibody-producing plasma cells. Importantly, to be fully protective, the T-cell-dependent humoral immune response needs to generate two waves of antibody. The first wave comes from short-lived plasma cells that have differentiated outside the germinal centers and that will produce low-affinity IgM. But to be fully efficient, the humoral immune response needs also to produce high-affinity mutated IgGs that are produced by long-lived plasma cells, and these cells can only differentiate inside the germinal centers. These two waves are important because if the best protection is offered by mutated IgGs, it takes time to generate these antibodies. And so the first wave of low-affinity IgM is important to protect the patient during the time that the response is maturing inside the germinal centers. An important question is to understand the mechanism driving the diversification of B-cell clonal progeny. Indeed, it's quite difficult to understand why these B cells, which are all the same, will choose to follow the germinal center pathway or the extra-germinal center pathway. A recent paper has shown that what was critical for a B cell to enter the germinal center dependent pathway and differentiate into long-lived plasma cells was the amount of T-cell help. The question is why B cells, which are resulting from the proliferation of the same mother cells, which are therefore identical, would receive different amount of T-cell help. We thought that this might be explained by the fact that the antigen was polarized inside B cells when the mother B cells start to proliferate. Now, this could lead to a different distribution of antigen between the daughter cells. To validate our hypothesis, we decided that we would analyze the distribution of the fluorescence of SPAG in mitotic B cells. These mitotic B cells are quite easy to identify using flow cytometry because they have twice the DNA content, so they are PR high, and they express phosphohistone 3. They represent about 1% of the cells in the culture at 72 hours. The problem is if you want to study the distribution of fluorescence in these mitotic cells, you need to analyze them by confocal. And this is when the trouble starts because you really look for a needle in the haystack. Of course, you can increase the percentage of mitotic cells by using nocodazole, for example. But because this drug is synchronizing the cells by interacting with microtubule networks, what you do is you may actually interfere with the very process you are trying to analyze. This is where I think the imaging flow cytometry is really powerful, because this technique really allows you to identify your mitotic B cells as easily as in flow cytometry. But in addition, because you can also study the distribution of fluorescence in your mitotic B cells, you can really have a clear picture of the different mitotic stages. As you know, the prophase is the phase where the chromosome assemble. So the prophasic cells, they have a round nuclei expressing pH3. In metaphase, these chromosomes are going to line on the middle plane. So you have only one nuclei that is flat. And in anaphase, one copy of each chromosome is dragged at one pole of the cells, and the two poles are going to become the two daughter cells. And they are quite easy to recognize because they have two flat nuclei. Not only can you identify these different stages, but you can also, all along the mitosis, track the distribution of the antigen, and you realize that the polarization of antigen is maintained across mitosis. Of course, the polarization of antigen across mitosis will lead to an asymmetric segregation of antigen between the two daughter cells, and you can quantify this by imaging flow cytometry. Indeed, as early as the metaphase, but also for anaphase cells, you can define the division plane, and it's quite easy using the imaging flow cytometry to calculate the amount of antigen of fluorescence you have on one side of the plane and on the other side of the plane, which allow you, for each cell, to calculate the division polarity index. Doing this for several cells, you can clearly see on the right-hand side of the slide that the division, the segregation of antigen between the two daughter cells is never symmetric. It's extremely rare that the two daughter cells will receive the same amount of antigen. Most of the time, one of the cells receives more antigen than the other, and sometimes one of the cells is receiving the total amount of antigen that was inside the mother cells. So here's this unequal distribution of the content of antigen into the two daughter cells, something that is actively driven. Is it related to asymmetric division, a process that we know, for example, is taking place in stem cells? As you know, whenever stem cells divide, the polarity machinery allows to distribute unequally the content of mother cells into two daughter cells. One will stay a stem cell, the other one will differentiate. And this polarity machinery is dependent upon several molecules, and one is particularly important, it's the peak acid zeta. So if the unequal distribution of antigen of mother cells into the two daughter cells was related to this active process of asymmetric division, we would expect that the unequal distribution of antigen would go along with unequal distribution of peak acid zeta. But using imaging flow cytometry, it's obvious here that the peak acid zeta is not unequally distributed, which suggests that the unequal distribution of antigen into the two daughter cells has nothing to do with asymmetric division. It has to be something that is driven by a different mechanism. So if it is not an asymmetric division that is explaining the unequal distribution of antigen between the two daughter cells, what is the explanation then? We think that it is related to the heterogeneity of antigen distribution in B cells. Because in fact, when you analyze mother B cells loaded with antigen by flow cytometry, it looks like it is a very homogeneous population as far as the antigen content is concerned. But if you use 3D electron microscopy to really analyze the distribution of antigen in endosomal compartments in this homogeneous population, if it is not asymmetric division that is responsible for the unequal distribution of the antigen content of mother B cell into the two daughter cells, what is the mechanism involved? We think it's related to the heterogeneity of antigen distribution in B cells. In fact, if you look at what looks like an homogeneous population of mother B cells as far as the antigen content is concerned, based on the flow cytometry profile on the left-hand side, you realize that these different mother B cells have distributed their antigen content differently in the endosomal compartment. If you, for example, pay attention to the cells in the middle line, it looks like all the spags are in the single endosomal compartment, while for the two other cells, the one on the top and the other one on the bottom, it's as if the spags were distributed in different endosomal compartments. Because the two mother cells contain the same amount of antigen, they will interact with T cell with the same efficiency because they will present the same amount of MHC class II molecules containing peptides. But whenever these cells are going to divide, they will lead to the generation of daughter B cells that are going to be different. In fact, if the antigen is distributed in several different endosomal compartments, then these daughter B cells will receive roughly the same amount of endosomes and therefore the same amount of antigen. This is the case that is illustrated on the top of the slide. In contrast, if a mother B cell contains the same amount of antigen, but this antigen is concentrated in a single endosome, because endosomes cannot be split between the two daughter B cells, only one daughter B cells will inherit the total amount of antigen that was inside the mother B cells, and this will lead to an asymmetric segregation of antigen, as illustrated on the bottom of the slide. What are the functional consequences for daughter B cells of keeping the antigen from mother cells or losing the antigen of mother cells? So to try to address this question, what we have done is that we have purified mother B cells that had the same antigen content. We have placed these mother B cells in culture with T cells. We have waited 72 hours so that the mother B cells have divided, and they have generated daughter B cells in red that have kept a high content in antigen, and daughter B cells in blue that have lost the antigen. And these two populations of daughter B cells have been sorted again and placed in culture with a new batch of T cells that were CFSA labeled. And what is interesting is that only the daughter B cells in red, the one that had a lot of antigen inside, have been able to trigger T cell proliferation, which was quite obvious, but which means that these daughter B cells in red have a better ability of the blue ones to interact with T cells. And now it's interesting because starting from the same clone of B cells containing the same amount of antigen, we have a progeny with different ability to interact with T cells. And so now we can understand why the same B cells can enter the germinal center because they receive a high amount of T cell help. It would be the red one, the red B cells, while some other, because they have lost the antigen when the mother B cell is divided, lose the ability to interact with T cells and are probably pushed to differentiate into short-lived plasma cells. Because the different daughter B cells, the green at the top of the slide and the blue and red at the bottom of the slide, have inherit a different amount of antigen from the mother B cells. They have different efficiency in interacting with T cells. And therefore, in that sense, we can say that it is the way the antigen is distributed within the endosomal compartment of mother B cells that decide the fate of daughter B cells that are therefore not created equal. So based on what I've just discussed before, the $1 million question is now, what are the parameters that are really influencing the distribution of antigen within the endosomal compartment of a mother B cells? Of course, we have seen previously that it was possible to study the distribution of SPAG in the endosomal compartment of B cell using 3D electron microscopy. But you need to realize that this technique is not really useful to study what parameters can influence this, because just to get this nice 3D picture of this single B cell took me 3 days. So here again, the imaging flow cytometry looks like the ideal technology to answer the question. Indeed, it's quite easy to follow the fluorescence of the SPAG inside the B cells and to use an antibody that is directed against the protein at the surface of the SPAG to determine whether the SPAG is inside the B cells or not. For example, for the cell that is on the top of the slide, only one SPAG is inside the B cells, and the other one, in contrast, is positively stained with the anti-OVA antibody, suggesting that the SPAG has not been internalized by B cells, and it's quite easy to follow the kinetic of internalization of this SPAG along the time. Imaging flow cytometry is very good at numerating the number of red spots you have inside the B cells, and all these spots, they correspond to endosomal compartment. And you can realize on these particular cells over here, that has 5 different spots, they have not the same size and not the same fluorescence, suggesting that these different endosomal compartments contain a different amount of SPAG. Now it's very easy to get on the B cells that have only one fluorescent spot, which means one endosomal compartment containing SPAG. And if you analyze the intensity of fluorescence of these spots in these different cells, you realize that you get different spike. The first spike is for the cells that have only internalized one SPAG in one endosome. The second spike is for the cells that have internalized 2 SPAG in a single endosome. And by plotting these numbers, you get a slope that gives you the quantum of fluorescence for one single SPAG. And now that we have, with imaging flow cytometry, the number of red dots in the B cells, and that we have also obtained the quantum of fluorescence for a single SPAG, we can use ImageE to analyze the intensity of each spot and to define the number of SPAG per spot. And this is a very, very efficient tool that provides you a high throughput analysis at a single cell level of a number of SPAG per endosomes. With all these numbers, you can calculate what we call the antigen distribution index, which gives you a sense of how much the antigen is concentrated in a few number of endosomes. The more this antigen distribution index is important, and the higher is the antigen concentrated in a few number of endosomes inside the B cells. So now that we have all these tools set up, we have decided that we would study the impact of the intensity of BCR signaling on SPAG distribution within the endosomal compartment of B cells. Why so? Why so? Because, as I told you before, it is the BCR signaling cascade that triggers the association of microsignalosomes with dynein. And this is the dynein that will drag the microsignalosome along the microtubules up to the microtubule organization center, resulting in polarization of antigen. So it makes sense to put forward the hypothesis that the intensity of BCR signaling will impact on the way the mother B cells will distribute the antigen into the endosomal compartments. So for this part of the study, again, we will use synthetic particulate antigen, but we have made small changes. Instead of coating the nanoparticles with an antigen, we have coated these nanoparticles with an anti-kappa antibody. And this anti-kappa antibody will bind to the BCR, mimicking the binding of cognate antigen to the B cell receptor. So to convince you that this trick is working, I'm going to show you that we can trigger BCR signaling, which leads to phosphorylation of SLP65, also known as BLNK, in B cells. So here, what we have done is that we have put in suspension purified B cells. So it's a mixture of kappa positive B cells, because the B cells, the BCR, they have two kinds of light chain, lambda or kappa. And because we are using an anti-kappa antibodies, only the B cells that have a BCR expressing kappa light chain will be activated by our antigen. So using flow cytometry, you can follow the level of phosphorylation of BLNK in lambda B cells, lambda positive B cells, so we have a negative control here, and kappa positive B cells in red. As you see, you have a shift in the phospho-BLNK signals. But it's not great, and I thought you do have a shift. This shift is really not great, and I think it illustrates quite well the limit of flow cytometry analysis. So here again, the imaging flow cytometry is doing better than conventional flow cytometry. In fact, the problem with flow cytometry is that you have a background signals that is making your life difficult whenever you are looking for the positive signal of phospho-BLNK. But if you use flow cytometry, you realize that the distribution of phospho-BLNK is different in kappa and kappa positive, and lambda positive B cells, even for the cells, where the fluorescence, the total fluorescence doesn't look so different. You realize that in the kappa positive B cells, the fluorescence is concentrated in a single spot, while in lambda positive it's more evenly distributed, suggesting that this is background. Even better, you can use the fluorescence of SPAG and analyze the colocalization of SPAG fluorescence in red and phospho-BLNK in yellow to demonstrate that it is actually the SPAG that are triggering the BCR signaling inside the B cells. And because it's very easy to do, you can do kinetic analysis showing that with time, the probability that the SPAG will trigger the phospho-BLNK signaling is going higher. So now if you consider a single type of SPAG with a certain amount of anti-kappa at the surface of this SPAG, they will trigger phospho-BLNK in B cell, as I've shown you before. But because whenever you put this SPAG on the B cell in suspension, not all the B cells will contact the same amount of SPAG. This will have an impact on the amount of BCR signaling you are triggering in these B cells. For example, here, the B cells that are in the red gate, they have bound a lot of SPAG because the fluorescence is high, and they, of course, have more phospho-BLNK signaling than the B cells that are also kappa B cells, but they have encountered less SPAG, and the phospho-BLNK signaling is lower. What is interesting is that you can change the composition of SPAG. You can increase, for example, the amount of anti-kappa you coat at the surface of SPAG, and this also will have an impact on BCR signaling, which is what we try to achieve actually. For example, here, in these different situations, you have on the top here the number of molecule of anti-kappa that has been coated at the surface of SPAG, so it goes from a bit more of 1,000 up to 8,500 molecule of anti-kappa. If you look at the B cells that are in the red gate, they have the same SPAG fluorescence, so they have bound the same amount of SPAG, but because the SPAG have a different amount of anti-kappa at the surface, the phospho-BLNK signaling in the different red, if you like, histogram are going higher with the increase in anti-kappa at the SPAG surface. And it's also true, of course, for the B cells that have bound less SPAG, which are in the blue gate. Actually, you can summarize this data on... In fact, we can summarize this data on this 3D graph here, where you have the number of beads. When it increases for the same number of anti-kappa at the surface of a SPAG, then the phospho-BLNK signaling is going up. And the same is also true for the same number of beads. If you increase the number of anti-kappa per beads, you also increase the level of BCR signaling. Now, this is very important because what I'm telling you here is that we have a tool that allows to deliver different amount of BCR signaling for the same amount of antigen inside the B cells. And if we use these tools, we can really now study what is interesting us. So what is going to be the impact of BCR signaling on the antigen distribution inside B cells? So we have compared two different SPAG. One is in red. These SPAG have 8,500 molecule of anti-kappa, so they deliver a strong BCR signaling. And the other SPAG are delivering a low BCR signaling because they have only 1,000 molecules of anti-kappa on the surface. So we have compared the antigen distribution index that I've presented to you before for B cells that have internalized the same number of beads, going from 1 up to 14, depending whether they were SPAG 8,500 or SPAG 1,062. And as you can see here, the antigen distribution ratio is higher for the B cells that have received a stronger BCR signaling, suggesting that indeed, in accordance with our hypothesis, BCR signaling intensity is impacting on the way B cells are distributing antigen inside the endosomal compartment. So to conclude, I would like to put our findings in perspective and try to explain what could be the meaning of this. Let's imagine that we have high affinity B cells that have internalized a lot of antigen, and that these high affinity B cells divides its antigen content symmetrically between the daughter cells. As a result of that, it will take a lot of time for daughter B cells to lose the ability to interact with T cells. Because of that, all these daughter B cells will keep a high level of interaction with T cells, and they will be promoted to enter the germinal center. In a sense, this can be seen as a good thing because it will generate a lot of long-lived plasma cells producing high-affinity mutated IgGs that are extremely efficient in protecting the patient and providing memory. But the problem is that by doing that, none of the B-cell progeny will enter the extra germinal center pathway, and you will not have the generation of the first wave of low-affinity IgM. And this means that the patient may die actually in the waiting for the high-affinity IgGs to be generated. In contrast, in the asymmetric model, because the high-affinity B cell has received a strong BCR signaling, it has distributed the antigen into a small amount of endosomes. As a result of that, it will lead to an asymmetric segregation of antigen between daughter B cells. And this will allow generation of daughter B cells with no antigen content. These daughter B cells lose the ability to interact with T cells and enter the extragerminal center pathway of differentiation, leading to a formation of short-lived plasma cells that will generate this low-affinity IgM that will protect the patient waiting for the high-affinity mutated IgG to be produced. Finally, I'd like to conclude on a more technical note, trying to maybe insist on the fact that imaging flow cytometry is really the method of choice whenever you need for high throughput analysis of rare cellular events or when topological analysis of intracellular events is required. So this is the final slide of my presentation. I'd like to thank you very much for your attention, and also to thank the people that helped me to gather this data, Alice Koenig and Antoine Sicard, my two PhD students, Sébastien Dusurget from the flow cytometry facility, and Christophe Chameau from the microscopy facility at the INSERM unit in France. I'd also like to thank Andy Philby, a close friend who introduced me to the imaging flow cytometry, and of course my former boss in postdoc, Facundo Batista.
Thanks, Olivier. That was a great presentation to kick off our webinar. I think you provided the audience with some great insight into the power of imaging flow cytometry for high-throughput immunological analysis. Before we move on to Chris's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box at the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. Let's get our last presentation underway. Chris, the floor is yours.
Well, hello everybody. My name is Chris Parris, and I'm an academic at Brunel University in West London, where we have about 20,000 students and a very active research program. For the last few years, we've been using Amnis imaging flow cytometry for the detection of DNA damage in human cells using foci assays. And I'm going to tell you a little bit about that work. The key points of today's talk will be first to tell you a little bit about our research, then talk a little bit about how we've used imaging flow cytometry to develop a good method for gamma H2AX foci quantitation. Then I'll go on a little bit to say a little bit about how we've used imaging flow to detect DNA repair defects in human cells. These would be inherited repair defects. Most recently, we've been able to acquire a new generation imaging flow cytometer with, where we've been able to develop more enhanced and better protocols with the multimagnification capability and something called extended depth of field, which I'll come to a bit later. This is all leading towards developing a diagnostic test for the determination of chemotherapeutic and radiotherapeutic responses in cancer patients. And finally, for the last few slides, I'll talk a little bit about how we're investigating chemotherapy drug resistance in ovarian cancer. So the background to our group is we are very interested in clinical and cellular hypersensitivity to cytotoxic anti-cancer therapy, namely cytotoxic chemotherapy and radiotherapy. We've also in the past identified novel DNA repair defects that are passed on in an inherited fashion that are associated with clinical and cellular hypersensitivity. So therefore we've done fundamental studies of DNA repair. And as I said earlier in the slide, we're really looking at the development of biomarkers for the prediction of cancer patient response, and we hope that the imaging flow cytometer will be central to many of these developments as we go forward. So looking at gamma H2X just briefly, it's a useful biomarker for DNA damage, particularly when you irradiate cells with ionizing radiation, which causes DNA double-strand breaks. Gamma H2X initiates and forms at the site of DNA damage. And what happens is the H2AX is a member of the histone proteins of the nucleosome, and it's subject to phosphorylation. And at the site of DNA damage, particularly a strand break or a double-strand break, many thousands of these phosphorylations will occur and cause a focus which we can detect immunologically in the cell. So if we look at a kinetic of DNA damage after irradiation, you see within a few minutes at time 0, straight after irradiation, you see the induction of DNA damage as measured by gamma H2AX. And then within a few hours, the repair of the double-strand breaks occurs and gamma H2AX is dephosphorylated and we lose the appearance of those foci. If you look at in situ immunofluorescence, looking at a number of nuclei, if you look at the left-hand side of the slide, you can see a nucleus that's been stained with DAPI that has not got any DNA damage, and it's completely clear. But then if you irradiate the cells, you can see the appearance of those little green or yellow foci, and that's where we've used an antibody against gamma H2AX to recognize the DNA damage. Now, if we move across the slide, as we look at the upper nucleus, you can see that DNA repair has occurred, and most of the foci have disappeared. But perhaps in a scenario in an inherited DNA repair defect where there is no repair, you can see that after a significant period of time, the foci are still retained. And we've been able to use imaging flow cytometry to investigate these phenomena. So the initial image stream, imaging flow cytometer that we were able to acquire from Amnis Millipore was the Amnis Image Stream Mark 1, simple configuration with a green laser, a bright field laser and a dark field laser, a single camera with 6 optical channels, and routinely we would capture at least 10,000 cells per sample. The multispectral imaging that we would see would be a bright field image as you would look down the cells in a light microscope on channel 1. We would then see gamma H2X foci in either green or red stain on channel 2, and nuclear staining with a stain called DRAC5 on channel 5. So this slide just contains details of the immunocytochemistry that we would use. We use antibodies recognizing gamma H2AX, the phosphorylated form. And there's options for a couple of secondary antibodies that you can use, which can be a phycoerythrin conjugate or an alexafluor-488 conjugate. And also the DNA stain that I spoke about earlier, which is DRAC5, which is a very reliable stain for identifying the nuclear region of the cell. In order to quantitate gamma H2AX foci, you use the IDEA software. which comes with the Amnis imaging flow cytometer. And you follow a simple set of instructions to identify first the number of single cells that you have, then cells in focus, and then you define a truth population of cells with different numbers of gamma H2AX foci that you identify yourself. And then this allows the IDEAS software to enumerate all of the foci in all of the cells in your sample run. So going in a clockwise direction, in top left we have the identification of single cells by regioning tool. Then moving over to the right, we identify cells in focus, which is very important because we're taking pictures of cells, and the cells might be in slightly different positions relative to the camera as they move down the flow cytometry core. And then just at the bottom on the left there, you can see a truth population of cells that is identified by the user, which has different numbers of foci, and this is used by the software to calculate the number of foci in the cells. Then we use the masking manager to quantify gamma H2AX foci, and you can see 4 panels of cells there. First, we have a bright field image on the top, on the left, and then in the middle, in black and white, you can see a cell with lots of little dots, and those are the foci. And then on the right, you can see the region of the nucleus, which has been identified by the DRAC5 stain. So the first thing we do is we use the mask manager to identify the nuclear region. You can see that coming up in that lovely light blue color. And then we ask the software to identify the spots or the foci or the DNA damage in the cells. And you can see that in the middle of the third panel with those little blue dots. And then finally, we use a little bit of Boolean logic to identify all of the foci in the nuclear region. And that allows us to create a feature that we can use for calculation of all foci in all of the cells during our sample run. On this slide, we can see what that actually looks like. We've got 4 panels of cells. So on the left, we see the image of the cells in bright field. And then moving over one column, we can see the red stain there, and that stain is increasing in intensity and punctateness, and that's increasing number of foci. And those foci are shown in black and white, masked with the little mask manager, the spot counting feature. And then on the right, we have the DRAC5 nuclear stain identified. And then we can start to generate some data. So first on the graph to the left, we have a little frequency distribution, which is showing all of the foci in all of the cells for any particular sample. And if we move to the bigger graph on the right, you can see here we've done our kinetic, where we've looked at foci in untreated cells, and then cells up to 24 hours after irradiation. Now, if you look at the black bars, that's the kinetic you will see in a repair normal individual. At half an hour, you see induction of foci and then a rapid decline, and that represents the repair of DNA damage. In the white bars, however, you see induction of foci and then persistence of the foci. And this is because in this individual, they have an inherited defect in double-strand break repair, because these cells are derived from an individual with ataxia telangiectasia, which is a classic inherited syndrome where there is a failure to repair double-strand breaks. So that's really interesting. We can show differences between normal and repair deficient individuals. Now in the grey bars there, you see kind of like an intermediate response. And again, this is a cell line from an individual, again with a defect in DNA double-strand break repair, but not as severe as the ataxia telangiectasia patient shown in the white bars. So very gratifyingly then, using the imaging flow cytometer and our gamma H2AX quantitation assay, we can identify subtle differences in DNA repair between different patients. In the last year or so, we've been able to upgrade our imaging flow cytometer to a Mark II with 2 additional features. We have the multimagnification capability, including a 20, a 40, and a 60x objective. And also, we've had a feature added to the machine called extended depth of field, or in other words, a focus stacking capability. So this allows us to project all structures within the cell into one crisp plane of focus. So theoretically, that should allow us to see more gamma H2AX foci in different regions of the nucleus. So we went back to our original experiments, repeated them to see if we could identify a difference in the number of foci that we were observing. And we did this with and without extended depth of field, EDF, using the 40 and the 60x magnification. And if we look at this next slide, we can see the pattern of foci induction that we get when we use our MRC5SV1 repair normal cells. Again, the white bars show the induction and repair of foci over a 24-hour period at 40x magnification. The light grey bars show when we add the EDF, so we're seeing more foci. And then the slightly darker grey bars show the 60x magnification, but then when we go to 60x with EDF, we see many more foci. So again, that's really nice. This enhanced magnification and the extended depth of field is allowing us to see more damage within the cell. So that's probably approaching in situ immunofluorescence techniques using an epifluorescence microscope. If we repeat the same experiment with our repair defective cell line from the individual with ataxia telangiectasia, which is the double-strand break repair defective cell line, we see induction again and then less repair when compared to the normal cells, but as we add the EDF and the increased magnification, we start to see more foci. So it clearly seems that with these additional features, such as the high magnification and the extended depth of field, we are seeing more damage and we're getting more information from our experiments. On this slide, we can see we've got some examples of what that looks like. Panel A, B, C, and D. And what we have here is 40x magnification, then 40x with EDF, and then 60x magnification, and 60x with EDF. And I think that you'll agree is if you look at panel B and D, you're beginning to see many, many more foci because you've got the extended depth of field switched on. It's making the foci look a lot cleaner and much easier for the analysis software to enumerate the foci. So it really is quite a nice feature to have the high level of magnification plus the extended depth of field. The question I'm often asked is, how does imaging flow cytometry compare to in situ microscopy using a 100x magnification objective? Well, we did that experiment where we looked at the number of foci in our repair normal cells, the MRC5SV1, compared to our ataxia cells and our XP cells, which are also defective in double-strand break repair. And if you remember from the earlier slide, we saw slightly different patterns of DNA repair. In the normals, you see the induction and the repair, and then in the repair defective cell lines, you see greater or lesser amount of repair. So the pattern that we see in this graph is identical to the previous graph that we showed, but if you notice the number of foci that are being identified with in situ techniques, at a maximum we're seeing about 30 foci per nucleus per cell. And that's very similar to the 60x magnification with the extended depth of field. So that's very gratifying. We're approaching in situ levels. But the really important thing for us is to take this information and using the imaging flow cytometer from Amnis Millipore is to develop a diagnostic test. Now, in my group, we're really interested in why and how patients respond to chemotherapy and radiotherapy. And we're really interested in this from the viewpoint of side effects. Many patients suffer quite severe side effects, others not at all. And this has been a question that's been looked at over a long period of time by oncologists, and it's been standardized into scales of side effects. And here is one called the Radiotherapy and Oncology Group Scale, and it's called NTT, and what we mean by that is normal tissue toxicity. So this can start at 0, classification 0, with no side effects, right through to group number 5, which is the case where when you have treatment such as radiotherapy, it can result in death. And that would be the case with our ataxia telangiectasia patient, who have no ability to repair double-strand breaks that would be induced, in this case, by the radiotherapy. But you get all of these different responses in between. And the ability to be able to predict this would be really important in terms of personalized medicine and reducing side effects, unpleasant and painful side effects that patients suffer. So how we did this, we took a liquid biopsy, 10 mils of blood from our cancer patients, identified the peripheral blood lymphocytes, or PBLs, by buffy coat, treated the cells with 2-grey gamma irradiation to induce the damage, and then did our kinetic of repair, and then we used flow cytometry to detect gamma H2AX. In normal individuals without cancer, you see, as before, rapid induction of strand breaks, and you can see the foci in that little image there just above the peak. But then after a few hours, you see a return to normal, and that represents in normal individuals that they're repairing the damage correctly. Now this slide shows something similar, and it's from patients that had cancer, but had very, very little side effects. They did not experience much normal tissue toxicity. And again, you see the induction of damage, a slightly slower repair, but by the time you get to 24 hours, the cells have repaired the damage. Interestingly, in patients that suffered these really severe side effects, what we see right across the board here, to a greater or lesser extent, is you irradiate their peripheral blood lymphocytes, the cells report the damage, but there is no repair. So it seems that our gamma H2AX assay is allowing us to differentiate between those patients that have very severe side effects compared to normal individuals, or cancer patients that experience very little side effects at all. So that's really gratifying. So if you combine all the data, you can see the normals, non-cancer individuals. The dotted line represents the cancer individuals that had very few side effects. And then there are the red line, the combined data, where all of these individuals had very severe side effects, so easily distinguishable. So the next thing is to validate this test. And we have to go through something in Europe called Conformity European Validation. And that meets a very strict set of conditions which will allow us to use this in a diagnostic setting. So the idea is that we can use this in routine radiotherapy clinics for the prediction of response to clinical radiotherapy, and hopefully individualize treatment on a patient-by-patient basis. Lastly, I'd like to talk a little bit about how we're investigating mechanisms of chemotherapy drug resistance in cancer patients. And we're particularly interested in ovarian cancer, in as much as there's about 7,000 cases in the UK of ovarian cancer. Survival rates are very low, 5-year, 40% survival rates, which is exacerbated by the fact that it's often diagnosed late, that these patients develop drug resistance to chemotherapy drugs. And there's lots of good evidence that drug resistance can be associated with enhancements of DNA repair. Central to chemotherapy protocols for ovarian cancer are the use of bifunctional alkylating agents such as cisplatinum. The cytotoxicity of these bifunctional alkylating agents is directly related to how well that drug causes an inter-strand cross-link, whereby the drug binds to both strands of the DNA. This is a very challenging adduct for cells to repair, and it requires the coordinated interaction of multiple DNA repair mechanisms. So the important adduct is highlighted in that little red box there around that second DNA damage. and that's the interstrand cross-link. We call those ICLs. As I said earlier, ICLs are very, very difficult to repair. We generally require the process of homologous recombination, where endonucleases cut out or unhook the damage, and a number of proteins are important in that. That we've got site-specific endonucleases, XBF, ERCC1. We could use gamma H2AX to detect the strand breaks once the endonuclease cut out the damage, or we could look at the Rad51 protein, because Rad51 is very important in collecting together all the various components of the homologous recombination pathway. And we hypothesized that in drug-resistant ovarian cancer cell lines, there would be elevated expression of these proteins and that this would be associated with drug resistance. So our first graph here is looking at the number of gamma H2AX foci in sensitive and drug-resistant cells after exposure to drugs like cisplatinum or a similar drug called nitrogen mustard. And essentially the white bars show the number of H2AX and Rad51 foci that we see in the drug-resistant cells over a time course after drug exposure, up to 72 hours we're looking at there. And you can see that we get more H2AX foci, representing more incisions of the damage, and we're getting a greater assembly of the homologous recombination pathway by the increased expression of Rad51. So it would appear that in drug-resistant cell lines, we see this increase in DNA repair. Just to take that a little bit further, when we look at our ovarian cancer cell lines, and essentially where we've got those very tall white and dark bars and grey bars, that represents the drug-resistant cell lines of ovarian cancer that appear to be cutting out the DNA damage more effectively than the drug-sensitive cell lines. So here we've been able to use the imaging flow cytometer to investigate issues of drug resistance in cancer cells. So to summarize, we're heavily invested in imaging flow cytometer for identification of DNA repair defects in human cells, and we've developed good and reproducible protocols for spot counting, such as gamma H2AX and Rad51. We're moving into identifying mechanisms of chemotherapy drug resistance in human cells, and our diagnostic test development for the prediction of radiotherapy and chemotherapy side effects is really important to us. And we would also add that the multimagnification and the extended depth of field brings imaging flow cytometry close to in situ methods. The advantage is that you get near the same number of foci identified, but you can count many more thousands of cells. So that gives you good statistics. So to finish off, there are my contact details, and it would be lovely to hear from anybody who might want to collaborate with us or to use our imaging flow cytometer for your sample runs, particularly where you're looking at DNA damage, but we've developed protocols for a number of other endpoints as well. There's a few selected references in this slide which might be useful to you, which we would invite you to look at. Finally, these are the members of our group: myself, my two postdocs, Emma and Sheba, my clinical fellow, Rachel Lewis, PhD student, Hussein, and my collaborator at one of the top hospitals in London, at St Bartholomew's Hospital, Dr. Nick Plowman. And finally, just a little slide on funding there. Thank you very much for listening.
That was excellent. Thanks very much, Chris. I think the audience is now keenly aware of the benefits for using imaging flow cytometry methods for improving DNA damage assay diagnostics. Before we start the Q&A session, I want to let everyone know this is your final chance to submit your questions for our speakers. We've gotten some really great questions already, but I'm encouraging all of you in the audience to keep them coming. All right. While everyone is submitting their final round of questions, let's begin the Q&A so that we can try and get to as many questions as possible. All right, guys, so let's get to the Q&A session. We have a bunch of questions that have come in, some of them really great. So the first question is going to be for Olivier. Olivier, one of our audience members would like to know, is the pH3 labeling required to separate prophase, metaphase, and anaphase stages, or can you do that with the morphology of the nucleus alone?
Well, it's a very good question. Actually, no, the pH3 is not necessary. We used to use this staining to make our life a little bit easier whenever we were trying to detect the mitotic cells, but it turns out that the pH3 is not very well expressed by anaphasic cells. So it's really good to detect the prophasic and metaphasic cells, but not the anaphasic cells. So we changed our mind, and recently we used another marker that is a MPM2 antibody that is more stably expressed across mitosis. But for people that would like to detect the different mitotic stages without labeling the cell, it is possible. And you should refer to the recent publication in Nature Communication by Parris early this year. And I really think that they have shown that it was possible using the imaging flow cytometer to detect the different mitotic stage for a cell using only the bright field image from the amnis.
All right, great. Thanks very much, Olivier. Chris, first question for you. Audience member would like to know, what other applications have you used in imaging flow cytometry?
Well, we've got a number of groups in our division that are now sort of getting invested and using the imaging flow cytometer. So we've got people that are looking at microparticle identification, which is quite interesting. We've got people that are using the image stream for things like NF-kappa B, intracellular transcription factor localization. I myself have also looked at apoptosis using things like an Xn5 labeling of cells. So those are some of the sort of interesting applications that we've used. We're also developing protocols, sort of an enhanced protocol for cell cycle identification. Now there is a wizard within the IDEA software of the imaging flow cytometer which does this. But we're trying to come up with a really simple protocol that you can actually do in live cells, and we'll hopefully be submitting something to Cytometry A very soon.
Great, thanks very much, Chris. Another question for Olivier. Olivier, how common is the uneven distribution of surface antigens after the cell division? Do hematopoietic stem cells exhibit this property of uneven distribution during the division?
Well, this question is quite difficult. As you know, there are a couple of papers claiming that cells can divide asymmetrically. I wouldn't be able to answer for whole cell type, but what I can say is what I've seen analyzing B-cell division. And regarding surface antigen, I have been unable to detect any whatsoever asymmetric segregation of surface antigen during B-cell division, but we haven't tried everything. We have looked for CD25, MHC class 2, CD86, and some other co-stimulatory molecules, and CD69, which is an activation marker. And all these surface markers in B-cells during the mitosis are evenly distributed between the two daughter cells. So that's pretty much everything I can say about that. I haven't, but that's
okay. Thanks, Olivier. That was great. Chris, question for you. Audience member would like to know, for foci identification from WF images, did you use deconvolution or not? It would probably affect the numbers as well as magnification and stacking.
Yeah, I mean, when we did the sort of in situ work, we just used simple sort of epifluorescence. We didn't have access to something like a confocal or a deconvolution capability. However, I guess if you did do that, it might enhance a number of foci that you can actually see, and you'll probably see a lot more foci. Essentially, with the extended depth of field on in the imaging flow cytometer scenario, you're kind of doing a sort of a mathematical deconvolution to pull all of the images that in the sort of within the depth of the cell onto one focal plane. But no, we weren't able to do that with the in situ type approaches, but that would be sort of an interesting analysis to do and to compare that with the extended depth of field on the imaging flow cytometer at 60x. Having said that, we think that we've got pretty good resolution of foci using the imaging flow cytometer at 60x with EDF. We're really quite pleased with that. And sort of rather than doing sort of a lot of this comparative type work now, which is interesting in itself, and this kind of work needs to be done, we're kind of pressing on with the development of some of these sort of diagnostic procedures, because we think it would just be really wonderful if we could take sort of a machine like the imaging flow cytometer and actually have it as a diagnostic platform, and that would be very exciting for us.
All righty, thank you very much, Chris. Olivier, looks like we have one more question for you. Audience member asks, in your time-lapse example, was this done by running the same sample at 1-minute intervals or parallel wells?
No, well, unfortunately, it's not possible to use the Omnis technology to do whatever you would like to do, or what you could do in a time-lapse microscopy. You cannot follow a single cell longitudinally. It's pretty much as with FACS analysis or flow cytometry analysis. You really need to run parallel samples. So that's what we did.
All righty, thank you very much, Olivier. And it looks like we have time for one more question for Chris. Chris, when you prepare your cells for imaging flow cytometry, can you use the same protocols for classical non-imaging flow?
Yes, well, when we started all of this, we didn't sort of really have a lot to go on there. But we did have access to protocols for standard non-imaging classical sort of flow cytometry, and that's where we started. And generally, we found out that many of those protocols were completely adaptable to the imaging flow cytometer. And we took it a little bit further. Sometimes we didn't even have a protocol that we could develop from the not from non-imaging flow cytometry. So we would go to in situ methods and say, right, let's just see if an in situ method would work here. And generally, you can find out that you can quickly adapt protocols. So it really is, in terms of cell preparation, it's really quite straightforward. I think one of the important things that we would recommend if you're getting into imaging flow cytometer is having a decent cell pellet. If you have a sample of cells and the pellet is very small, by the time you've gone through so many iterations of staining and then washing of the cells to wash away excess antibody, you always lose a few cells on the way through. So a good number of cells as a starting point will ensure success that you can have a decent number of cells that go into the imaging flow cytometer when you do your sample run.
All right, great. Thanks, Chris. And with that, it looks like we've come to the end of our webinar. So I'd like to remind everyone that our webinar will be archived on our site for 6 months at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward it to friends and colleagues, which we always recommend. I'd like to thank Olivier and Chris again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Millipore Sigma for sponsoring this webinar. So hopefully we'll see you again in another Gen webinar in the near future. Goodbye for now.